Good evening to you all. I'm Kanyerezi Stephen, and I'm presenting a study titled Characterizing Microbial Diversity and Resistome in Urban Sewage using shotgun metagenomics in Kampala, Uganda. My supervisor is Dr. Gerard Mbowa. So our general objective is characterizing microbial diversity and resistome in urban sewage using shotgun metagenomics in Kampala, Uganda. Specific objectives: 1, we have to identify and characterize antibiotic-resistant bacteria in urban sewage in Kampala. And the second one is to determine the urban sewage resistome. Resistome meaning the totality of resistant genes within the sample. So first, antimicrobial resistance is a cross-cutting and increasing threat to global health. This undermines the progress that is taken towards the treatment of infectious diseases. And for the current surveillance of antimicrobial resistance, is usually targeting a few pathogens only, and this is based on passive reporting of phenotypic laboratory results, which cover only the human clinical infections. This procedure leads to significant time delays and incomparable data and a narrow pathogen spectrum. So this leads to capturing of low antimicrobial resistant genes, which tries to undercover the actual antimicrobial resistant genes in the samples. From a surveillance point of view, urban sewage is actually attractive because it provides sampling material from a large and mostly healthy population, which otherwise would not be feasible to monitor. So our problem statement is sewage comes from different areas: hospitals, industries, and households. Then it joins a good drainage system, which transports the sewage to the wastewater treatment plant, which recycles, and this water goes back to the water bodies, that is rivers, lakes, and so on. So even given that it goes through this good system, this well-established system, but there's also some problems, as you can see from my screen. Here we have problems that are posed by the roots. Sometimes the pipes may wear out, leading to spillages to our environment, hence contaminating it. And remember, these spillages harbor antimicrobial resistant bacteria. So our focus, we are going to use shotgun metagenomics, which surveys the entire genomes of all the organisms present in the sample. As you can see from my slide, genomes from the sample, the DNA from the sample will be extracted, fragmented into short pieces, and sequenced. Then it will undergo a bioinformatics based analysis approach. So first of all, the ethics statement and sample collection. So our approval will be from School of Biomedical Sciences Institutional Review Board at College of Health Sciences, Makerere University, Uganda. And the samples will be collected during both the wet and dry seasons from a wastewater recycling plant in Waise before being recycled back. The DNA will be extracted using the Kaigen toolkit, then fragmented and sequenced using the Illumina sequencer. From this slide, you can see a sample output, how the data will look like from the sequencer. This will be our FASTQ file. So from that, we shall do a quality assessment, and we shall check for the quality of the reads using FASTQC. And then for the poor quality reads, and even those that don't reach a threshold of 25 thread score, they will be trimmed off using Trimomatic. Then we shall do a species identification using Metafaran 3, as you can see from my screen. Then we shall categorize the strain using meta-MLST, which categorizes the strains based on locus sequence typing of alleles. Then we shall do a de novo assembly using megahit. The contigs from megahit will be merged using CD hit est. Then from here, we shall use those contigs to predict the genes within the samples. Then we shall use abricate to determine the antibiotic resistant genes within the sample. Thank you. I acknowledge my supervisor, Dr. Gerard Mbowa, Makerere University IDI and the African Center of Excellence. Thank you.
Excellent. Thank you, Stephen. Are there any questions from the audience? In the meanwhile, I have one. I'd like to know, Stephen.
Yes.
Okay, I'll just go to Natasha's first. She says, "Why did you not use targeted sequencing rather than shotgun sequencing?"
Well, we are not only targeting, because for targeting sequencing we are going to only target that is the 16S. But here we are also interested in other organisms, that is, say, the viruses also.
Okay. And from George, he asks, "Do you expect to find a lot of novel species?"
Yes, novel species, I don't think we might find a lot. There might be one or two, but what I think it's about the novel resistant genes is what we are expecting to find.
Okay. And then Natasha then asks, "Will you look into the microbiome, the fungus?"
The fungus, yeah,
Yes.
because given that this is not just a targeted sequencing, actually this will help us do that too.
Okay. Alrighty, great. Thank you so much, Stephen.
Okay.